Good morning, everyone. I'm Kamila Muchowska, and I'm coming to you from France, from Strasbourg. I'm a postdoc in the lab of Joseph Moran, who's my advisor at the moment. And I'm going to talk to you about something slightly controversial, I would say, still for some, which is maybe a little bit sad at the moment. But, well, now for something completely different, let's talk about the origin of life on Earth and whether metabolism can emerge and function without enzymes. So us as humans, we've been fascinated by the mystery of the origin of life since we could think. So to trace the origin of life back in time, we've arranged the history of life in this pyramid of complexity. So at the bottom, you have the least complex things. At the top, you have the most complex organisms, including us. So this pyramid presents to us some sort of a chicken and egg conundrum, because, okay, this is not working. Okay, well, can you hear me from here? I'm gonna shout, all right. So we have metabolites here, which build enzymes. Metabolites. Okay, now it's better. So metabolites function in metabolic pathways, but metabolic pathways are catalyzed by enzymes. Enzymes themselves are made from metabolites, which are on the lower rung. However, how these enzymes are made is encoded in genetics, which are even more complex. So if you want to trace the origin of life back in time using this order of complexity, this becomes a little bit difficult. So we must ask ourselves a question: how do we progress from simple geochemical inputs and environmental inputs to something just a tiny little bit more complicated, such as metabolites? And by metabolites, I understand simple molecules such as acetic acid, alcohols, even glucose. These are all fairly simple. So the area I work in is called Prebiotic Chemistry, and the role of this area is to try to explain how we could progress from geochemistry, right here, which is an abiotic world without life, to this complex biochemical map, which presents lots of pattern and lots of organization. To help us understand which kind of biochemistry could have emerged as prebiotic chemistry, it's very helpful to make two fundamental distinctions about how life is organized. So first distinction is whether organisms are reductive or oxidative, which means whether they gain energy by taking electrons from the environment or by releasing electrons to the environment. Another distinction is whether they are autotrophs or heterotrophs, which means if they have to hunt and kill other animals or others, prey on other species to gain energy, or do they contain the whole metabolism within a single organism? So several scientists, as these guys presented here, these are evolutionary biologists, chemists and biochemists. They have done some studies, and for now we think that the major amount of hints about the origin of life comes from reductive autotrophs. So now about the geochemical scenario. Reductive autotrophs, but we are on the early Earth. So how does it fit? We know that the core of the Earth is full of reduced metals, such as iron and nickel. Then if we progress towards the outer areas of the Earth, through the mantle and to the crust, we encounter more oxidized minerals, minerals that contain iron +2 and iron +3. What you end up with could be compared to some sort of a giant electrochemical Earth battery. So think of the Earth as a giant battery. This can provide a lot of energy. So those organisms that I mentioned, reductive autotrophs, they build up biomolecules from simple carbon inputs, for example, CO2. There are two pathways in which which they use that are the most evolutionarily conserved. The first pathway is called the Wood-Jungle pathway here. Basically, to cut the long story short, you take 2 molecules of CO2 and you obtain acetate, which has 2 carbons. Okay, the second is a bit more complicated. It's called Krebs Cycle, but in reverse. You might remember Krebs Cycle from high school. The reverse Krebs Cycle uses acetate up here, then by adding carbon molecules, it ends up as a citrate, and then the citrate is split. For non-chemists in the room, if you don't understand anything about chemical structures, the smaller the squiggle, the simpler, the bigger the squiggle, the more complex. So we're going for the least complex to the more complex. What is important about this Krebs cycle is that it contains 5 universal metabolic precursors to all of biochemistry that we know. So from acetate you can obtain lipids, from pyruvate sugars, from oxaloacetate aspartate, and also nucleic acids. From succinate you can obtain cofactors. From ketoglutarate you obtain glutamate and other amino acids. So this is a hub of biochemistry, 5 universal metabolic precursors. How this works? Well, there is a hypothesis that once on Earth these 2 pathways were fused and they operated together. Well, actually, in actuality, what I told you before, on the early Earth there were no enzymes because enzymes are complicated by default. So is it possible that this existed without any enzymes whatsoever? You could think it's crazy because CO2, many chemists in the room, we know CO2 is not very reactive. You need catalysts to make it do anything. But actually, many of the enzymes that catalyze these pathways, they contain metallic centers in their enzymes or use cofactors that are metallocofactors. So if you look at the green blobs, these are symbols of metals that promote all these reactions. So the question for us was, can we use those metals alone to do these reactions in the laboratory? Well, let's see. This is what we do. For non-chemists, a quick explanation. We perform reactions in little vials. We perform a parameter screen, which is called high-throughput screening, and then we analyze. For analysis, we use 2 machines, standard in chemistry, NMR, and GCMS. What it looks like, it's here. You fill up quite a large amount of little tubes with different reactions, different parameters. You put them in pressure cookers. These are pressure cookers. We pressurize them at high pressures. This is safe. My colleague from the building can confirm that we evacuated the building only once over 3 years, so it's fairly safe. And yeah, that's what we do. For the first pathway, where from 2 molecules of CO2, we wanted to obtain acetate. In one of the screens, we found that by using metallic iron, just metallic iron powder from Sigma-Aldrich, we could promote the conversion of 2 molecules of CO2 to acetate, and even better, adding another molecule of CO2 to the first step of the RTCA cycle, the reductive Krebs cycle, to pyruvate. This reaction is very robust over a large temperature gradient and over a large pressure gradient. And what is cool, from these 2 graphs, major conclusion, many temperatures, broad range, and also it works at 1 atmosphere of CO2. So you don't really need to use the pressure cooker, we found after the fact. Okay, the next step was trying to tackle the cycle. That was a bit more complicated because there are many reactions, so we divided the cycle in 2 halves, the bottom sequence and the top sequence. So both sequences contain the same type of reactions. The pink reactions are reductions, for which you need electrons. The green reactions are reactions where you either add or remove water. The pink reductions are simple. Many reductants work. Iron powder was a good reductant, so we used it. The green ones were a little bit more complicated because they are all equilibrium. So it's hard to make them work on their own because they flip back and forth between the 2 products. So after screening with my colleagues over 4,000 reactions, we did it quickly. We had a good master student from China. And so eventually we managed to come up with conditions that could promote both sequences in one pot, 3 steps. You don't need to purify after each step. So this is in contrary to what many organic chemists do to elucidate this mystery. So they claim they perform prebiotic chemistry, but they have to change conditions after every step. They have to purify, run a column, and then only then can they proceed to the next step. We run both sequences. So I mean, from oxaloacetate, reaction number 3, 4, and 5. So you plug in oxaloacetate, you end up with succinate. That's the bottom sequence. Under the same conditions, oxalosuccinate, reaction 8, 9, and 10, you end up with citrate. So 2 sequences under the same type of conditions, acidic conditions, quite compatible with prebiotic settings. And they are also compatible with the previous linear pathway that I showed you from acetate to pyruvate. So to summarize quickly, that's not the end though. We managed to find common conditions for a large part of the hybrid pathway proposed by many evolutionary chemists as the origin of life pathway that could have given rise to protein metabolism that works without enzymes. For this, we use iron, we use zinc, and we use chromium ions. We can use up to 35 bars of CO2, but we don't have to. You can use 1 atmosphere of CO2 and it's still fine. Temperatures, I put 100 degrees here, which is sort of the common ground for both type of pathways. The first one can work even at room temperature. In the cycle, the bottom sequence can also work at room temperature, but it doesn't have to. The upper one works at higher temperature, and those conditions agree. What is missing here? So reactions that are missing in the red arrows, red dotted arrows. So this loop, reaction 2, reaction 6, reaction 7, and 11. These are all the reactions that need ATP. ATP is the biological energetic currency, and it's a complex molecule. It contains phosphate. So what was so special about reactions that need ATP to be performed by biology that we couldn't, we just couldn't do them? So the next thing we tried to look for, okay, maybe in the beginnings of life, there was no need for ATP. Maybe there was a portion of metabolism that functioned completely independently of ATP. So recently there was a paper in Cell by David Gray and the team where they took essentially the whole entire KEGG database that contains the entire biochemistry known to humanity, and they removed all the reactions that needed ATP, removed phosphate. And what was left was this. These are all the reactions that can function in biochemistry without phosphate. So it's still quite a robust network. If you think about it as of a metro map, there are some hubs which are more well-connected than others. There are those little hubs and nodes that are more well-connected than others. So the black circles I put on this map are the 5 universal metabolic precursors that I showed you on the Krebs cycle. And they are quite well connected, but not necessarily. The most well connected ones are these, pyruvate, and the red one is an extra molecule. It's a glyoxylate molecule. So what does it mean? Glyoxylic acid, pyruvic acid, why are they so well connected even though you don't need ATP? We tried, what we did was to put them in together in a vial with water with some iron, +2 ions, and what happened? Okay, this is a very, very fresh finding. It's been on bioarchive, on ChemArchive since Tuesday. since Tuesday, it has been, at the moment, there's a conference in Munich about the origin of life going on. This paper has been online since Tuesday. I just got a Slack message from my boss yesterday who said 3 people at the conference in Munich have already talked about this paper, which has been online for 3 days. So this is quite a hot thing, so hold on. So what happens if you put glyoxylate? There's an error in the structure, by the way, it must be a chemdrop bug. So what happens if you put glyoxylate and pyruvate together? Under these conditions, you wait 1 hour, you get all this. So this clearly must mean something, like 2 molecules that are the most well-connected molecules on the metabolic map without ATP. If you put them together in water, very mild conditions, they give rise to a huge complexity, which really, really resembles the Krebs cycle. You need 4 reactions extra, and you will have a complete Krebs cycle. So perhaps it's not so difficult for metabolism to have arisen before even life was considered life. And this is what I want to leave you with, a take-home message, is that life and geochemistry, geochemical inputs are very, very well connected. You need iron, iron salts, warm water, some salty water, and you can obtain much of metabolism that you, like the reactions of which you can see in biochemistry today. If in this short talk I made you a little bit interested in this type of research, please have a look online. We have 2 blogs we've written for the Nature community about these topics. And with this, I would like to thank several people. First of all, I would like to thank you for giving me the chance to speak here. And I want to thank the group I work with. They are all amazing folks. My supervisor, the postdoc advisor, is a great mentor. Most of this work I did together with this guy, Srijit. He just defended his PhD thesis days ago. And I thank you for your attention.